Hello? Oh, I got it. Okay, so let's get started. So we're on time. For the next three talks they're on POGZ. So the first one we have Dr. Eirene Markenscoff-Papadimitriou from Cornell University, who's going to tell us about the functions of POGZ.
Thank you very much. I have to say it's really humbling and a great learning experience for me to be here and to meet the families affected by POGZ and CHAMP1. And I'm really learning a lot, and it's really benefiting the research in my lab. So POGZ was first identified around more than 10 years ago as a high-risk autism risk gene from whole exome sequencing studies that were done by the Simon Sequencing Consortia. And now the Autism Sequencing Consortia has sequenced more than 20,000 individuals, and 102 risk genes have been identified. And POGZ is still at the top of this chart. And so the striking thing about these genes is that many of them regulate other genes, which makes it very complicated to understand. So what are the genes that are actually important? And in this talk, I will be talking about POGZ, ADNP, and CHAMP1 is also on the list. So subsequently, more and more individuals with deleterious variants in POGZ have been identified, and now it's recognized as white Sutton syndrome. That's we've heard a lot about this with intellectual disability, delays with language, autism-like behaviors, gastrointestinal and vision problems. And so POGZ is very important for development, and so as researchers who are molecular biologists and neuroscientists, what we want to understand is we've never heard of these genes before up until recently. We want to understand, what do these genes do in the teeny tiny cells of our body? What are their molecular functions? How do genetic variants in these genes affect the ability of these genes to do what they do? And how can we repair their functions to rescue the phenotype? So I'm going to talk in my presentation, I will talk a lot about epigenetics. So what is epigenetics? You might have heard that the DNA in the cells of our bodies can stretch all the way to the moon. And so the way that the cell manages to package all this DNA into the nucleus is through these necklaces of chromatin. And so the DNA is wrapped around these proteins, these beads, that allow the DNA of our cells to be packaged within the cell's nucleus. And this is actually a very dynamic process by which some DNA is active at some stages in development, and then it becomes silenced, or vice versa. And so what we're interested in understanding for POGZ and CHAMP1, which we know associate with these necklaces in the cell nucleus, is how do mutations in these two genes affect the dynamic nature of chromatin during development. So to study, we use genetically modified mice to introduce a POGZ mutation. And we used CRISPR-Cas9 to target exons 1 through 7 of the mouse genome for POGZ. And this generated a deletion allele of POGZ in mice. So once we generated this mice, we observed that they had a very striking behavior. When you would open the lid of the cage of the mice would jump out of the cage, and then they would jump off the bench onto the floor. So these mice seemed fearless, and so they seemed like they had reduced anxiety. And so I gave them to a collaborator at UCSF, who put them in this maze. It looks very similar to the maze that Mary Matos showed you earlier with an elevated circle maze. Here it's an elevated plus sign maze. So usually well-type mice don't like to go into the exposed arms of the maze because it's scary. It's up high from the ground and it's exposed to the air. the mice prefer to spend time in the closed arms of the maze. And so here you see the wild-type mice walking along the closed arms of the maze. And what we observed with the POGZ heterozygote mice is that they spend twice as much time in the open arms of the elevated plus maze. So they have reduced anxiety-like behaviors. And then we did recordings from brain regions that are involved as the mouse is walking around the maze and deciding where to go next. So as we recorded from the prefrontal cortex and the hippocampus, and usually wild-type mice, as they approach the center of the maze and they need to decide, are they going to go to the open arm or the safe closed arm, there's increased synchrony in oscillations between these two brain regions. These two brain regions sync up their activity patterns. And in the POGZ deficient mice, this synchrony was decreased. So it seems like these two brain regions are not communicating effectively as the mice are walking around the elevated plus maze. And so for us, we are very interested in understanding how do the molecular mutations that we're studying affect the ability of these two brain regions to communicate and for the neurons to connect with each other. So then we study the homozygous knockout for POGZ. So in these mice, these are sections of mouse embryo brains. So if you just take a slice of the brain, it will look like this. And you can see POGZ in purple and in red is highly expressed in the wild type. And in our homozygous knockouts, where 2 copies of POGZ have been deleted, POGZ is no longer expressed highly. And so we did RNA sequencing to discover what are the genes that POGZ is controlling during mouse brain development. And we found around 150 genes go up, 150 genes go down. The genes that go down are very important neuronal development genes. These are 2 genes, SLITRAC1 and SLITRAC5, and these are, now the purple is expressed just in the part of the brain that becomes neurons. And in the knockout, POGZ homozygous knockout, these genes are no longer expressed. The same here. These are very fascinating genes that are important for neurons to form connections with each other. SLITRAC1 is a Tourette's disorder associated gene and also associated with obsessive compulsive behaviors. Latrophilin 3 and FLIRT2 are part of this fascinating complex that forms at the cell membrane of neurons. And Tom Sudoff at Stanford, Nobel Prize winner, has discovered this complex. And FLIRTs are presynaptic and latrophilins are postsynaptic. And depending on which flavor of FLIRT and latrophilin interact, the cells, neurons either repulse or attract towards each other. And so, as you can see in our POGZ knockouts, these genes are no longer, there's no purple here in these brains. And so what we think might be happening as a result of POGZ homozygous deletion is that important genes that control the patterning of the brain and neurons' ability to connect with each other are being seriously impacted. So how does POGZ do this important thing to regulate genes that are important for neuronal development? We used a method called cut and run. I won't get into the nitty-gritty technical details, but this is a molecular method that allows you to isolate embryonic mouse brain cells and use an antibody to find where is POGZ binding DNA. And so with this method called cut and run, we see that POGZ binds around 2,000 different locations in the genome. And these locations are overlapping with other signs of regions of the genome that are active, that are euchromatic. I talked a bit about how DNA is packaged in the genome. POGZ is binding loosely packaged DNA in the genome. And so what this analysis shows us from the POGZ knockout is that POGZ promotes the ability of neurons to grow and send off their axons and form synaptic connections by binding loosely active regions of the genomes called enhancers to regulate nearby genes. So POGZ is a complicated protein. We're really trying to understand what aspects of its many functions are contributing to white Sutton syndrome. One of the aspects of its function that we're interested in is its ability to organize DNA in the cell nucleus. So the cell nucleus, DNA looks like a bowl of spaghetti, and the spaghetti isn't actually randomly organized. So you can actually map the interactions of one strand of a spaghetti to another strand of spaghetti. And so what we think POGZ might be doing is organizing the strands of spaghetti so that it's not a random mix, but it's actually highly organized. So what we're doing, the experiments that Tasha, who is here at the meeting, is doing is she is analyzing embryonic POGZ knockout mice and dissecting the cortex, this developing part of the brain, cross-linking cells, and doing experiments called micro-C. Micro-C allows you to map the strands of spaghetti within the cell nucleus and map the connections between the different strands. And so this is a representation of Tasha's data, where in these pyramids are these pyramids indicate that the base of the pyramid is interacting and touching within the cell's nucleus. That this part of the strand of spaghetti and this part are actually touching. And so what Tasha is seeing in the POGZ knockout cortex is that these interactions that seem to be consistent from cell to cell are falling apart in the POGZ knockout. And these interactions, these pyramids, are only falling apart at the loci of these important neuronal genes that we've mentioned. So SLITRAC1 and SLITRAC5 is highly expressed in developing mouse brain and is not expressed in the POGZ knockout. And here we see the interactions of this locus are lost in the knockout. This data is so beautiful, so I couldn't resist showing you more that the pyramids are also disappearing at another gene that's very highly expressed in brain development in the wild type and is lost in the knockout. Okay, so it gets more complicated with POGZ, it always does. So POGZ binds a protein called HP1, and HP1 also binds another autism risk gene, ADNP. And so we wanted to see what fraction of the parts of the genome that POGZ controls is also controlled by ADNP. And so we did cut and run for ADNP together with HP1 in the mouse embryos, and we see that around half of POGZ's binding sites, 1,000 different locations in the genome, are co-occupied together by ADNP. So it's possible that these two genes have overlapping targets during mammalian brain development. Another thing we found, going back now to the heterozygote mouse, is that we saw that one of the top genes that is downregulated in the heterozygote is ADNP. And by this experiment, you can see in purple is ADNP in a wild-type mouse, and you see that there's less decreased purple staining in the heterozygote. And it was just recently that we've started to appreciate that POGZ and CHAMP1 form a molecular complex, or a machine, as I recently heard it referred to, within the cell nucleus. And so we are doing the same kinds of experiments using homozygous CHAMP1 mice, and you can see CHAMP1 expression is beautiful and matches what we see with POGZ. And so we are repeating these same experiments, and Tasha and Eirene, who are both here at the meeting, can tell you more about their experiments trying to replicate these same results with CHAMP1 mice. Okay, so where do we go with POGZ and CHAMP1 research? We are interested, like we did for POGZ and ADNP, to identify common targets for POGZ and CHAMP1 by catching these proteins in the act of regulating the same DNA sequences and the same genes. And then we want to identify which are the brain cells that are deficient in these genes. It's not going to be every single brain cell in which these proteins are acting. And so we want to identify what are the neuronal subtypes which are deficient in these genes, and then maybe try to boost these cells and their connections in the brain and the expression of these downstream targets in these. And we're very excited to do these experiments in mice. This cartoon here shows that I believe you're going to hear later today that CHAMP1 really is important for regulating this tightly packaged chromatin. But what we see with POGZ is that it's also regulating active chromatin as well as heterochromatin. And so we're very curious about the overlaps between these two circles. So just as with CHAMP1, we have the issue of that we heard mentioned about haploinsufficiency versus dominant negative. And we're also modeling truncating mutations in from POGZ patients. And we have generated a mouse model where a single nucleotide deletion in the splice acceptor site for the final exon of POGZ generates a truncated POGZ. And so we're very interested to really parse out what are the contributions of the different domains of POGZ to these effects on chromatin and gene expression. And this is a really elegant experiment that's relevant to the patient, to understanding this patient's mutation. So all the research I've showed you so far is our mouse modeling of POGZ, which is very powerful because we have a mouse, it behaves, and then we also have the mouse embryonically is generating all the different cell types and is expressing all these really important genes that pattern the brain and its connections. But we also want to use patient cells and patient cell lines, like what we've heard previously with organoids or iPS-derived neurons, to really understand how the different genetic variants in POGZ and CHAMP1 affect the POGZ-CHAMP1 interaction. So, for example, we believe that mutations in this domain of POGZ might affect the ability of POGZ to interact with HP1 protein and CHAMP1, and vice versa. That mutations in the more C-terminal domain of CHAMP1 might be affecting the ability of this protein to interact with POGZ. Okay, so I mentioned that Tasha and Andrea are doing beautiful work in POGZ and CHAMP1 mouse models, and your participation in this research is really important, and if you're interested to learn more about how you can be involved, please reach out to me or to the Simon Searchlight team that's here at the conference. I'm happy to take any questions.
Thank you very much. You lost me at the reduced anxiety because anxiety is like the main thing we deal with. Mm-hmm. So my question is, do you, and maybe this is a dumb question, but do you study the mice at like different ages? Because, for example, our daughter, when she was 4, we didn't really see that much anxiety, but at 16, that's the biggest thing we deal with.
Yeah, I honestly, I don't think there's a way to compare mouse anxiety to human anxiety. So we studied them in adults. So we did not look earlier on just to see about developing mice anxiety. But Yeah, it could be, and then we tested like social anxiety. There was no effects for social anxiety, so maybe it could be something specifically about this maze and the anxiety induced by this particular situation. Yeah.
Sorry, very nice work. Have you considered either doing like a double knockout of POGZ and CHAMP1 or overexpressing ADNP or other downstream targets and seeing if you get rescue of the micro C chromatin
Yeah, that's definitely,
interactions?
it's a lot of work, unfortunately, for the two students sitting right next to you, but yeah, that would be, yeah. That's a really wonderful idea, yeah.
So is there a mouse model or a human syndrome for ADNP?
There is, yeah.
And is there
Van der Aa syndrome, yeah.
Are there phenotypic similarities with CHAMP syndrome and WSS?
Yes, definitely.
So it's a pathway. It's a pathway.
It's a pathway, yeah.
So maybe it'll show up in the AI pathway analysis that we heard about this morning.
Yeah. So we're able to pull down ADNP and HP1 with POGZ. We haven't tried yet for CHAMP1.
I'll ask some questions there.
That's very correct. Yeah. So I think like across development, we're able to sort out the neurons as they're differentiating in vivo, and so we need to really be paying attention to what stage in these cells development, because early on it could be acting by itself, and then later on it teams up with ADNP and CHAMP1 or vice versa. So it's really important to map the dynamic nature of these interactions.
Sorry for doing the housekeeping. We do have also remote listeners, so please use the microphone always, otherwise we are getting crazy.
Beautiful work and very impressive. So we stained for POGZ in the brain, and we actually saw differential subcellular localization depending on the type of neurons, I guess, and I'm not an expert in neurons.
Yeah.
So I was wondering whether that's something that you have seen also in your brain tissue, and whether you could maybe correlate POGZ.
Yeah. You see different subcellular
like
or subnuclear localization.
Subcellular. So actually, sometimes we see POGZ in the cytoplasm.
Wow.
In certain subtype of neurons,
Mm-hmm.
and I can probably show you all the picture if you want, because this is something that we're definitely not an expert in. While normally you would see POGZ in the nucleus, and it was very striking when we did it. So I was very wondering if you saw that also in the mouse.
No, we haven't seen that. I would love to see what you've learned about that.
Thanks for a great talk. Thanks for explaining it so well for all of us. You said that in the mice you completely delete the POGZ gene. How do we compare then the results in the mice with people who have the POGZ gene still but a variant of it? How does that all compare?
Yeah, so it doesn't compare. The mice are die before birth, and so we're restricted to studying just what POGZ is doing during brain development, and then yes, it's very possible that the patient mutations when they're heterozygote will not have the exact same genes be affected, or we might need to use more sensitive approaches to detect them, because it's more harder to pick up than in the homozygous animals.
Yeah. Very nice talk and really beautiful work. To kind of touch on the point about the anxiety-like behavior, I think it's also important, for at least for the parents, to see that we call it anxiety-like behavior, but part of that is because of the assay was originally characterized by the ability of an anxiolytic to make a mouse spend more time in the open arms. We don't really know that it's anxiety at all. In fact, I think Fragile X mice, for example, show the same thing. They'll spend more time in the open arms. The MEF2C mutant mice, I'll talk about later, do the same thing. but it doesn't necessarily correlate with anxiety behavior in the patients. And I've really started thinking about it more as threat detection, threat vigilance, approach avoidance behavior. Maybe, and a mouse is not a human, as we know, and obviously standing out on the open arm, they're a predated animal, so they're worried about things above, et cetera. So there's reasons why they're in the closed arm. But I wouldn't my advice to the parents, at least, is don't get too hung up on the term anxiety.
Yeah.
It is a behavioral difference that we can measure and detect, but it does not necessarily correlate with what you'll see.
And I learned a lot from the session yesterday from the White Sutton families that vision impairment is a common problem, and it could be that this behavior is that these mice just can't see so well and they head out to the wrong part of the maze. So I just really I got this interpretation only yesterday from hearing about these issues. So like Chris said, don't get hung up on the anxiety.
I have a final question. Do you expect there to be an episignature for POGZ and CHAMP1?
What do you mean by that?
Like, so other neurodevelopmental disorder like CHD2 and I think Rett syndrome, they have a episignature that you can identify that are pretty robustly identified in the patients, and I think they're able to find it just using blood. Would you think the changes into the genomic landscape may potentially lead to an episignature that's identifiable?
So we haven't found one, but I think you're going to hear about one later from the analysis of LCLs and the effect it has on H3K9 trimethyl.